I just wanted to quickly remind you of the various presentations that we're going to have over the next 6 weeks or so. As seen up here, the presentation today is by Dr. Jonathan Page, which will be followed by 4 other presentations on the topic of medical cannabis. And Dr. Philby will be speaking in approximately 2 weeks from now, followed by Mark Ware and the Honorable Anne McClellan and Dr. Kilmer. So I encourage you to attend or visit and listen to as many of these presentations as possible so you get a complete overview and a better understanding of the implications of medical cannabis. 2 of the presentations, the last 2 that you see here, are going to be at the downtown Harbor Center campus. And as you can probably tell from the topics that we're going to cover, which is the task force recommendation report and legalization, that those sessions are almost completely full. That doesn't mean that you can't sign up for them. Please go ahead and do that. We're making arrangements for overflow accommodation for individuals. So we're already looking at 300 to 400 people coming to these events downtown. So don't let that hold you back. Go ahead and sign up and we'll make sure we can accommodate you. And again, this is the website for those of you that want more information about the topic, www.sfu.ca medical marijuana. And as I said, this is going to be live streamed as we speak. Word about our sponsors. We did receive a lot of support from various organizations that are listed here. And once again, we do appreciate the support, both from within the university as well as external too. It allows us to bring the speakers in to host a number of events. And those of you that have been here the previous lecture, please stick around afterwards because we have food and other bitings and drinks outside in the loggia. So please feel free to hang around, socialize, and meet new people. So as I mentioned today, the talk is by Dr. Page, Jonathan Page. Many of you know him. He is the president and chief executive officer of Anandia Labs, which is located at UBC, and he's also an adjunct professor at UBC. And he'll talk about some of his work with the cannabis genetics and cannabis genome. He previously was at the Plant Biotechnology Institute in Saskatchewan, and I'm reminded to tell him that the weather here in Vancouver is getting close to what it probably is in Saskatoon right now. Prior to that, Dr. Page had a number of other positions in Europe, and recently is on the board of directors of the Canadian Consortium for the investigation of cannabinoids. And he's originally from Vancouver. As you can see, his PhD and bachelor's degree were from UBC. Those of you that have followed Dr. Page's contributions, you'll know that he was one of the members of the group that actually sequenced the cannabis genome, and that was published back in 2011. There's been work since then on deliberating the genetic structure of both cannabis species, marijuana and hemp. Jonathan's worked on a number of other enzymatic and biochemical elucidations of things that happen in the trichomes. Those of you that are aware of the trichomes, that's where a lot of the molecules, in fact all of the molecules, accumulate. And prior to that, he's also worked on hops. As you'll see today, there's a bit of an overlap with hops. So please join me in thanking Dr. Page for coming here and agreeing to talk to us about developments in the cannabis genome. Jonathan?
Great. Well, thank you, Zamir, for the introduction and also the invitation to speak today. You know, sort of heartening for people who work on cannabis that SFU has sort of taken this risk. I was really impressed 2 weeks ago to see the president of a major Canadian university you know, opening this colloquium and also, I guess, dreaming about medical cannabis. Is that, I mean, in terms of the sort of credibility and validation of a subject area that's controversial like cannabis, having SFU getting behind it is really heartening and quite substantial. It sort of shows you where things are in 2017. So my interest in this plant comes as a plant biologist and as a plant biochemist and someone who's gone from plant biochemistry into plant genomics as well. And that's what I'm going to focus on in this talk. So it hits on some of the high points of the biochemistry that we did. So I'll hit some of the high points on the biochemistry, which may cause some of your eyes to glaze over if you're not sort of oriented to that way. But you know, I think it's an exciting plant anyways, and this kind of reductionist sort of dissection of the plant and its function really helps us understand how it fits into, you know, as a medicine or a societal interest. Cannabis is a multi-use plant that humans have been using for millennia. So it's both a source of food in the sense that it's seeds in hemp seeds. And you know, you can go into a health food store in Vancouver and pick up hemp hearts and hemp granola bars and hemp cereal, etc. So humans have probably been cultivating cannabis as a food source for thousands of years. Maybe even longer was its use as a source of fiber. So cannabis stalks, whether they're marijuana or hemp, have this high quality bast fiber, which is a kind of a phloem fiber associated thickening that is really it's like a living rope. If anyone, and there's probably not too many people have gone to a hemp field and tried to haul one of those plants out of the ground, but you're essentially trying to pull a rope out of the ground. And the roots will come up more easily than you'll snap the stalk off. That's because it has this high quality fiber. And that was used, you know, recently in rope, but probably was one of the reasons humans started using cannabis as a plant, because you could make twine and fishnet and rope and things useful for hunting and gathering food. But the interest that I have in cannabis is not so much as a source of seed or fiber. It's really that it's a drug and a medicine. And it's kind of unique in the plant world in the sense that, you know, we have lots of food plants, you know, whether they're, you know, wheat or rice or lettuce. And we have, you know, some plants that are still grown for fiber or sort of structural properties, you know, even trees that are grown for fiber for pulpwood. And some plants that are medicinal plants, but really nothing that's all three at once. And cannabis is kind of unique in that way. The molecules that give it its properties as a drug or a medicine are things like THC, the main psychoactive cannabinoid, and we'll delve deeply into the biochemistry of that compound. This is actually a picture that comes out of a paper that Ethan Russo, who spoke here 2 weeks ago, published in 2008. And it shows a shaman's grave in Western China. And that shaman was buried, or potentially a shaman, was buried with a bag or a container with something like 750 grams of dried cannabis flowers. And that grave was dated to about 3,000 years ago. So people were using cannabis for its drug purposes for thousands of years. And even, you know, the beginning of last century, the pharmacopoeias in the US and England and Canada included cannabis extracts, were sort of prescription drugs of the time. Just to talk about, you know, going from shaman's graves of 3,000 years old to 2017 in Canada, cannabis is still going strong. And in fact, if you open the newspaper, it's doing even better, you know, and especially if you're a stock investor, I guess, too. So we're seeing, you know, hemp being cultivated in Canada as a legal crop since 1998. There's about 85,000 acres, 35,000 hectares or so grown annually, mainly in Manitoba, Saskatchewan, and Alberta. And that's all for seed, or 95% of it is grown for combining as a seed crop for oil or for hemp seed products. And then medical cannabis has been authorized since 2001, and we have about 140,000 federally authorized patients in Canada, far more who are also using cannabis in a medical sense from dispensaries. There's about 36 LPs or licensed producers under the federal system, and they're growing in these, you know, large indoor, sometimes greenhouse production facilities. You can see those numbers are ramping up too, and this is what's driving this sort of stock market increase of the LPs, is that there's many, many more patients being signed up now than at the start of the current program in 2014. We're also looking at legalization, and with Anne McClellan and Mark Ware coming in. Mark was the vice chair of the legalization task force, and Anne McClellan was the chair. They'll be talking more about the process, but it's kicked off, and it began in 2016. I mean, the task force was working all last summer and into the fall. So that's where we are in societal sort of connections. But I'll talk about the plant as a plant in its botanical sense. So cannabis is a member of the Cannabaceae family. That's a single, a plant family that only includes cannabis and humulus, and I'll talk about hops in a bit. Zamir mentioned hops. Hops is the plant that's used for flavoring beer, so it's the bitterness of hops that give beer its bitterness. The genus is Cannabis. The species is debatable, though I tend to use in my work, I call it Cannabis sativa, a single species, and I worry about all the forms and subtypes and the rest of it. Beneath that, I don't talk about Cannabis indica or Cannabis ruderalis, though there is some debate there. It probably originated and grew naturally first in Central Asia, but humans quickly took it from that site of origin and spread it eastward to Southeast Asia, where it's a drug and a hemp crop, and then across Europe, and then with trade and colonization, it spread to Africa and to South America and to North America eventually. It's an annual herb, so it means it grows and sprouts, grows and dies in a single growing season. It's dioecious. That's a sort of somewhat unique property, but cannabis is not the only plant that's like this, that has a female plant and a male plant. They're separate sexes. And it's actually the female flowers, the buds, that are what are smoked as marijuana, and I show a dried bud there. So it's all about the female plant when we talk about commercial marijuana production. It's wind pollinated. This is the male plant shown here with this sort of yellowy floral structure. We have male plants in my lab at UBC, and they release so much pollen. It's just clouds of stuff, and it's tiny, very fine little pollen that goes everywhere. And you can see in like a hemp field in Saskatchewan, when the wind blows, you get these clouds of pollen produced as well. And it's diploid, which is so the Genome Organization. As I mentioned, Cannabaceae as a family, as a botanical family, is composed of only these two genera, Humulus and Cannabis. So Humulus, again, is the cultivated hop. There's hundreds of cultivars that are grown, and if you go into a craft brewery these days, you can see Citra hops and Centennial and Fuggle and various things. They all come from Humulus lupulus, which is the cultivated hop, and there's a couple of sort of other. Japonicus is a plant that's grown but not for flavoring beer, and then a kind of weird hop plant called Unionensis. Cannabis is really that single species sativa that I think sort of is the overarching organization in terms of the classification of the plant. But there's thousands of strains, and in hemp sensors, many cultivars are registered varieties. So there's a lot of variation in there. The Cannabis sativa would encompass hemp types and marijuana types. And I guess I'll take one step back too before Ethan hears me and sends me an email or something where I'm calling this plant marijuana. Maybe I should do sort of air quotes marijuana. It's just, I also agree. I tend to use the word cannabis in general and medical cannabis for medical marijuana, but it's sort of shorthand for drug type plants to use marijuana. So I'm not trying to be pejorative or anything when I use that term. It's kind of easier. So turning to that drug type, cannabis produces this mixture of bioactive metabolites. So I mentioned THC before. Cannabinoids are the main drugs in cannabis. THC shown here, CBD shown here, and there's a bunch of other related cannabinoids that I'll talk about shortly. And they co-occur with terpenoids or terpenes in the plant. Terpenes were the subject of Ethan Russo's talk mainly. They're the volatile sort of flavor, smell components, but they also impart bioactivity. And this sort of metabolic cloud of materials, and that's, I guess it's a bit of a pun, but it's also, the fact that there's this mixture of things in the plant, they combine to give it its effects. So both the psychoactivity, this euphoric high for which cannabis consumption is used for, but also pain relief and reduction in nausea and reduction in spasticity, etc., where there's actually good medical evidence for some of these properties. And so, THC is the main psychoactive ingredient, and it's likely the main driver of pain relief, but it's the mixture of things that sort of give cannabis the full effects. It's kind of the whole plant system that does that. So the cannabinoids as a sort of a focus would be a group of approximately 100 metabolites, and people are still digging out new cannabinoids, and chemists are isolating things that are present in very low amounts but are structurally novel. They're found only in cannabis. So there are compounds that would be sort of cannabinoids, that they would bind cannabinoid receptors in our nervous system, but structurally they're different. So we're going to call, these are the cannabinoids per se. They're in the biochemical sense, they're prenylated polyketides, sometimes called terpenophenolics. And these are the major ones. These are the ones that are sort of at present in higher levels in the plant and or have some biological function. So THC, cannabidiol or CBD are the two major players. Cannabicromene or CBC is present in cannabis strains. I'll show a chromatogram in a second that illustrates that. Cannabigerol is the decarboxylation product of cannabigerolic acid, which is actually the precursor to THC and CBD. THCV is a side chain variant. You can see there's a pentyl or 5-carbon side chain to THC and a propyl or 3-carbon side chain to THCV, and that side chain difference imparts very, very different biological activity to those two compounds. And then cannabinol is an oxidation product of THC, and it's present in like old hashish samples or bud that's been stored for 3 years at the back of someone's cupboard or something where there's been oxygen working away on it. And it, as Ethan said the last lecture, it has about a quarter of the psychoactivity of THC, but it has this sort of mild sedative properties. So those are the kind of major players. If I was going to say, give you guys a test at the end, that's like name all the cannabinoids that you should really pay attention to, these 6 would be them. The terpenes, on the other hand, which co-occur with the cannabinoids, are not unique to cannabis. And you know that just by the fact that pine trees smell like pine because of the presence of alpha-pinene, this one. They're very diverse, about 40,000 potentially different terpene structures. They're found in all plants. And I mean, sometimes they're called terpenoids or isoprenoids. I call them terpenes because in cannabis at least, there's not that much oxygenation of these. And they're composed of the monoterpenes, which are 10 carbon molecules, and sesquiterpenes, which are 15 carbon molecules. And these might be the sort of main players: myrcene, limonene, linalool, alpha-pinene. And then beta-caryophyllene was again talked about in the last lecture. These are the sort of ones that show up at the highest amounts. And this kind of analysis that I show in this slide illustrates this. This is data that comes out of my analytical lab at Anandia Labs, and I didn't put names to these strains that were analyzed. It's kind of generic data. But what we're doing here is we're using HPLC, or in our case, ultra-high pressure liquid chromatography, so a soft separation method that separates out cannabinoids. And this particular strain is sort of a little bit different than what you might get in the recreational context, in that it's a mixture of CBDA and THCA. And there's something to be said right there, is that the plant doesn't make THC or CBD. The plant makes the acid forms, so they're carboxylic acid forms of each of those. And it's only when it's smoked or vaporized or heated, in the sense of cooking a brownie in an oven, you lose the acid groups and you form THC. So this strain has, that's approximately 1-to-1, though the ratio is a little bit different. And then we see CBGA, or cannabigerolic acid, as a little bit blip, and some cannabicromanic acid, which is the acid form of cannabicromene. And that's kind of typical. You see lots of cannabicromene, and its pharmacology is a little less clear than the other ones, but it probably is a little bit like THC. And if we take a different instrument in the lab and then analyze using gas chromatography, we look at the terpene composition, we see a range of monoterpenes, things like myrcene often being the dominant terpene in cannabis. And that myrcene is also present in hops, and if you smell it, you would say, well, yeah, it sort of smells, if you know what marijuana smells like, it smells reminiscent of that kind of material. And these other compounds kind of modify the smell and the odor of that. And then there's things like smaller amounts of beta-caryophyllene and alpha-humulene. But there's lots of little blips. It's a real mixture of unusual terpenes that differ a lot between strains. So that chemistry, that would be just a snapshot of the sort of metabolic diversity that's present in cannabis. That's one strain, the terpenes and cannabinoids, but you can have, ones that are just almost pure CBDA, pure THCA, higher ratios of CBGA, and then all sorts of different permutations on terpenes too. And that's why, cannabis is so, it's interesting as well. It's maddening for physicians because they're like, well, I'm trying to prescribe something standardized and consistent to patients. People go into a dispensary in Vancouver and they're like, hey, that's cool, there's something different. It smells like lemon rather than pine. But the cannabinoids, so we've known those molecules exist in cannabis. We've known their structures for a number of years. It was 1964 when THC was first identified as the active drug in cannabis. But when I started my work more than a decade ago, this was the question we were asking, which was, how are the cannabinoids synthesized? What are the enzymes and the biochemical processes by which the plant makes things like THCA? And you can see that's the acid group I was talking about, again, on all these molecules. So one of the things we know about cannabis and cannabinoid production is that cannabis doesn't produce cannabinoid just in the leaves. It produces them in the flowers. And this is a picture that some people criticize me for because it's not the nicest looking marijuana plant in the world. But it does show you the female flowers developing. I took this picture as a postdoc in Germany so many years ago. And you can see the stigmas. Those are the white structure, the surfaces that receive pollen for pollination on the plant. But these sort of speckly structures on the leaves, on the floral tissues, those are glandular trichomes. And those are actually the site that cannabinoids are formed in and stored in. And if we look a little bit, well, so there's a stalk and then this kind of glandular head. If we look a little bit more closely with an electron micrograph, you can see that stalk and this kind of, almost looks like a balloon or something that's been inflated there. And that's essentially what's happened, is that we have and this is probably one of the best pictures we have on cannabis trichomes. It was taken by a postdoc in my lab, Tegan Colacini. And it shows the stalk, and you can see the reddish structures in these epidermal cells are chloroplasts. So that's green tissue, and then the stalk ends, and you get this kind of disc of cells. It's labeled here, the secretory cell disc. This is really the site that, the group of cells that they're not photosynthesizing. You're not seeing any chloroplasts in there. They're actually receiving sugars and nutrients up the stalk from the rest of the tissue, or rest of the leaf tissue. They're just completely devoted to making cannabinoids and terpenes, and then secreting them, pumping them into what we call the secretory cavity, which is, what you get is on the surface of all plant leaves in the epidermis, you get a sort of a plasticky skin called the cuticle, and it's there to prevent water loss and protect the plant and stuff. These cells, they'll synthesize and then pump the cannabinoids and terpenes underneath that sort of plasticky skin, and it kind of expands like a balloon, and you get this resin droplet that forms above the secretory disc. And that's sort of the biochemical factory of the cannabis plant. And cannabis is not the only plant species that has trichomes. Mint plants, basil plants, all these sort of aromatic plants that we use also are lavender, are making and secreting compounds into trichomes. But cannabis it's got some of the biggest, nicest trichomes, and clearly there's a lot of chemistry happening there because of, you can see the levels of cannabinoids in marijuana types might be 25, approaching 30% by dry weight. All of that chemistry is happening in trichomes. So what we said was, if we're trying to answer this question, how do you make cannabinoids? You go to the site of where the action is, and this is sort of like, you want to know where cars are made. You go to a car factory. You might go to Detroit or maybe Mexico now, I guess, but and you try to figure out how that process works. And so what we did was we took a kind of a fancy blender called a bead beater in the lab and took flowering cannabis of a high THC variety. And again, you want to sort of give yourself the best chances of getting at the sort of signals you're interested in by focusing on high THC and then selectively breaking apart trichomes and filtering off the secretory cell discs and then isolating RNA. And RNA, ribonucleic acid, is the sort of the messenger that is used from the genome to create protein. So it's a kind of it's like, sort of like the working blueprints of the cell. So we isolated secretory cell clusters, got RNA from them, and then started sequencing the DNA form of those RNAs. And this gave us a gene expression catalog that was, well, it turned out to be very representative. It was kind of the Rosetta Stone of the cannabinoid pathway, and that over time and through a number of now published papers, we found these highlighted genes that would encode enzymes involved in cannabinoid formation. So like the most abundant, and contig size represents how many RNAs were present in our library at this point, related to polyketide synthase. It's involved in the sort of central part of the cannabinoid pathway. THC acid synthase was present. Olivotolic acid cyclase works with this tetraketide synthase, this aromatic prenyltransferase. So we when I say we figured it out, it was like 7 or 8 years of work and everyone in Sunday afternoon and all that kind of stuff. But we got there, and I just wanted to sort of highlight one example. So I could give you my biochemical talk for 45 minutes, but I'll just take one example. This is the sort of textbook biochemistry of how cannabinoids are formed. So you start with a short chain fatty acid called hexanoic acid, and you activate it, you add a CoA ester to make hexanoic-CoA, and then the polyketide synthesis step is this one. You take hexanoic-CoA, malonyl-CoA, and you make olivotolic acid. And then olivotolic acid, you add a 10-carbon geranyl group to that, you get CBGA, which is actually something that shows up in cannabis when you analyze it. And then CBGA is the substrate for THC acid synthase, cannabidiolic acid synthase, and Catamacromanic acid synthase. So I just wanted to show you, sort of how we got at the aromatic prenyltransferase. So this enzyme here, we knew that, we were looking for something that would add a geranyl group. Yeah, that's by design. I'm just sort of highlighting the enzyme. Sorry. Yeah, I could go back and maybe it won't strain your eyes as much. But I just wanted to focus on this one. It adds a geranyl group. And so what we did to discover that particular enzyme is we took that Rosetta Stone gene expression catalog and we looked our way through it till we found an enzyme that was, or a gene that was annotated as doing something that was biochemically similar to the cannabinoid pathway. In this case, it was vitamin D biosynthesis. And we said, well, that gene and that enzyme that it forms is looking like it should do something close to transferring an aromatic or a geranyl group. And so what we ended up doing was expressing that gene in this case in yeast, taking the recombinant protein out of yeast and actually doing the assay. So we took the assay where we put olivotolic acid and geranyl diphosphate, and we actually saw that the enzyme could make CBGA. So this was a sort of an example of using this trichome-focused genomics to discover enzymes, and then using that approach, we laboriously walked through the pathway, again, 6 or 7 or 8 years, where we identified the hexanoic-CoA synthetase, these two enzymes, the aromatic prenyltransferase, and these two, the THC acid synthase and cannabidiolic acid synthase, were actually identified by a Japanese group using a sort of different technique, but some similarities. So we were sort of almost fully able to complete the cannabinoid pathway using that trichome genomics focus. CBCA synthase sort of remained unknown because it's not actually found to be expressed much in trichomes, and I'll talk about it more in a few minutes. So all of those sort of trichome-focused genomics and using that as a resource for biochemistry got, was a lot of success, and there was a lot of nice papers out of it, but it also wasn't everything the plant does. I mean, the trichomes are only just one small group of cells, and the whole genome, which is really all the blueprint of the cannabis plant, wasn't known. And I was on sabbatical in California in 2009, 2010, and I actually someone forwarded me an email saying, hey, and it was from a University of Toronto professor named Tim Hughes, and he said, I think sequencing prices are down to an extent where we can do the cannabis genome using, bits and pieces of grant money. And he was really interested in cannabis because, it has biomedical properties, and nobody had touched what was clearly the most fascinating plant on the planet. So we started collaborating. We put together like a Canadian team that was the first to sequence and publish the cannabis genome, and it came out in a paper in 2011, and Zamir referred to that in his introduction. Whenever I give talks, I always check. This paper has been accessed something like over 100,000 times, so people love it. It's used by many grad students in their seminar courses to talk about genomics in a cool plant. What we ended up sequencing actually was we focused on Purple Kush, which was a variety or a strain that's still widely available in the medical cannabis world. It was grown by a friend of mine in Vancouver at the time, who was a MMAR licensed grower, and therefore he could serve as a source of DNA for the sequencing project. So although we did this big sequencing project, it probably cost about $150,000 or so to do the sequencing and lots of work to do the assembly. It was really hard to get government okay to get access to that DNA, and I ended up going to a patient, actually a guy who was in a wheelchair and growing material for his condition to get that DNA, and we acknowledged the growers in that paper. Since that time, I've been able to get that same strain back in my lab, and it's kind of like, it's kind of the lab rat of the cannabis genomics world. So we sequenced Purple Kush, and that's a high THC medical cannabis strain, though used potentially recreationally as well. And then we did some comparisons to a couple of hemp varieties that were grown by Canadian farmers, because we wanted to sort of figure out some of the differences between hemp and drug-type cannabis. Cannabis, I sort of show a bit of a family tree of sequenced plant genomes, and this figure is a little bit old, but cannabis sort of fits into a part where strawberries, cucumbers, apples, peaches, etc. It's not particularly different. I mean, we think of cannabis as kind of this unique status in the plant world. Its genome is pretty ordinary, other than the fact that it has genes that make cannabinoids and terpenes. And the size of the genome, 820 megabase pairs, is kind of middle of the road. It's a little bit of a difficult genome, and certainly we need a better genome. The genome that we produce is a good resource, but it's not a done deal by any means. But it's got a lot of repeats, a lot of transposable elements and things that make it kind of complicated to do the work. So one of the things, when we sequence a genome, we got a nice paper, we put the data out publicly. It's still on a server at the University of Toronto, and it was submitted to the US-based NCBI GenBank. So we wanted that resource to be widely used by the scientific community, especially with people who couldn't access the plant themselves, but maybe wanted to dig into evolutionary questions or other biochemical questions, where they could then just publicly start or sort of querying our sequences. And so that's been a resource that's been, I think, widely used. The paper's been cited a number of times. Some of the things we've used it for, though, are more biochemistry. So Catamacromanic acid synthase was out there as an unknown enzyme. And as I point out here, CBCA is present in medical cannabis strains. So it's not a sort of irrelevant molecule to be thinking about. What we did, if I go back here, was we said, if CBCA synthase takes cannabigerolic acid as its enzymatic substrate, and it's doing a reaction that's reminiscent, at least, of the formation of THC acid and cannabidiolic acid, the enzyme itself must be pretty similar to the known ones that are THC acid synthase and cannabidiolic acid synthase. So we Jake Stout, who was a guy in my lab at the time, looked at all of the close relatives of those two enzymes, THC acid synthase and CBDA synthase, and we came up with a list of a number of them, including things that were very, very close to THC acid synthase. So this would be a little sort of family tree where this is CBDA synthase, this is a non-functional CBDA synthase that another group in Japan found, and then a couple of other relatives. This one was very close to THC acid synthase. In fact, so close that the computer kind of collapsed their sequences together when we first looked at them. So Jake ended up expressing a number of these candidates, and he actually ended up finding that this homolog 5 would make Catamacromanic acid. So this was an example of using this new genome resource as a way of extending our ability to sort of probe functionality and find biochemistry. So the core cannabinoid pathway is complete, and that was lots of hard work, but I'm one to often say, there's not enough science done on cannabis. We need more science. There's so many questions out there. We certainly put that in grant applications and things, but we also have lots done. These are lots of papers and lots of information is there. Turning now to the sort of some of the differences that we get in the cannabis plant. So marijuana and hemp, and again marijuana because that's drug type cannabis, are the same species. They've been selected for different uses. So there's been lots of human involvement in cannabis. This has led to over, I think, a longer period, a sort of focus on fiber and seed in some forms, and then more recently, a real push to increase THC levels in drug forms. Again, hemp is this low THC type, while drug type is high THC. And that's kind of like a biochemical switch, if you will, that what we have is, I was showing you that pathway in the sort of vertical orientation before. This is horizontal. Cannabidiolic acid is taken by either one of these enzymes and made into either CBDA or THCA, and this would be sort of a purple kush type marijuana plant, a big peak for THCA and nothing really for CBDA. And then a hemp type would have a peak, a less amount of CBDA and maybe a little blip of THCA. So there's this kind of like, in the domestication and selection of the plant, there's been this attempt to make marijuana more potent, but also select for one single cannabinoid, and that's THCA. And in fact, that's led to a kind of a classification system, and this is data that comes out of me just taking public information about the medical cannabis strains that were available from licensed producers for Canadian patients or made accessible to Canadian patients in 2015, where I took that data and I said, okay, what is the percentage of THC and the percentage of CBD that they list? And then we see, a large number of high THC strains that contain virtually no CBD. And that's the vast majority of medical cannabis types that are available in the market now in Canada. And those would be called, there's kind of a classification system, and I think Dr. Russo referred to it, they would be called chemotype 1, pure THC types with maybe a little trace of CBD. And then you have chemotype 2, which were sort of like that chromatogram I showed before where it was CBDA and THCA both there. And then and those are those kind of one-to-one mixes of the two of varying potency. And then you have chemotype 3, which are really high CBD and very little THC. And those seem to be like really the two or the three types that are possible. And one of the questions is, why is that really the only type? Like, why wouldn't you get something out here? And there's a couple of ones that didn't show up. They're actually purposeful blended mixes that one of the licensed producers, Cannabis makes by mixing CBDA and THCA containing plants in here. So one of the reasons is that it's the genetics that determine these cannabinoid ratios. So what you can get in some plants, which would be drug type plants, chemotype 1, high THC levels, it's because you only get functional THCA and the gene for CBDA production is inactivated. It's a null. There's a bit, there's a little bit of leakage through, so maybe there's a tiny bit of functionality. And then chemotype 2, you're getting activity of both of those genes. They're making enzymes that will take both. And the two enzymes will actually sort of compete for the same substrate, the same food source. And that's sort of why if you have, if you had a strain with 20% THCA and you crossed it with a CBDA plant, you're not going to get 20% THCA and 20% CBDA, in total 40%. They're going to split it. They're going to be 10 and 10 and 9 and 11 or something. And that's what's happening in chemotype 2. And chemotype 3, which is really hemp type, you're only getting the functional cannabidiolic acid synthase, and that's making high CBD plants. And that's sort of what's legal as a hemp type, with Health Canada saying you can't have hemp growing commercially in Canada with more than 0.3% THCA. So you're really confined to these sort of really almost non-functional THCA production, and nobody's going to get high off 0.3%. So the regulation of cannabinoid production, to put it in a sort of slightly less biochemical terms, is you've kind of got a light switch. You've got this THC acid synthase. You've got a middle setting, I guess, which is chemotype 2, which is both of them, and then a third setting, which is CBDA synthase. And then you've got this dimmer switch, which is why hemp is 4% CBDA or something, 4, 5, 6% CBDA. And cannabis, marijuana types might be 26, 27%. And that's high. I mean, most stuff is around 20, 22. It's because, as well as that switch, you've got some sort of regulation. And we're still trying to work that out. So we know the genetic basis of the light switch, but the dimmer switch is still a question. And that's more, questions around, is this pathway flux, which is just kind of like, is it all about the pathways like that give the precursors, that give the building blocks to make cannabinoids? One of the things we think, and this is based on comparing marijuana types, purple kush, and hemp types, which in this case is phenola, is that we see much higher expression, and I don't want to get into all the names, but things like CSPT1 was that aromatic prenyltransferase that I talked about before. So it's more highly expressed in the purple kush than it is in phenola. And the THC acid synthase, of course, is more highly expressed in the purple kush, while CBDA synthase is the other way around. So there seem to be generally just more of these genes turned on in marijuana types. And we think this is to do with the fact that human selection, and this is an unusual kind of breeding program that wasn't done by Pioneer Hybrid or Monsanto or what have you. This was a breeding program to achieve high THC levels that was done in basements and gardens in Northern California and in BC and the Kootenays and in Amsterdam, where people were combining their sort of expertise and exchanging seeds and all aiming to sort of breed better and better cannabis, and better, until at least recently, typically meant higher THC levels. And that process was all about kind of increasing the expression of the cannabinoid pathway, probably also increasing the number of trichomes and the size of the trichomes to achieve these high levels. I don't know, I don't have my watch. How I do time-wise? I'm okay. The other thing that we've been using that genome information for is trying to piece together trying to make sense of some of the classification of the plant. So as I mentioned before, I'm kind of a believer in this cannabis sativa model where we've got one species and all of the other variation, when hemp, marijuana, strains of marijuana, indicas, whatever, can sort of fit beneath there. And it's possible that there were sort of subspecies at some point that were relevant. Or there's some competing views that would actually say there's other species mixed in there. There's Cannabis sativa, Cannabis indica, Cannabis ruderalis, which is kind of a weedy sort of ditch form, feral type form of cannabis. In many plants, we have, organized varieties. So if you go to, the grocery store and you find a Macintosh apple, you basically know it's a Macintosh apple. There's been this sort of horticultural registration system. Tomatoes, there's a lot of diversity, but you can order packets of tomatoes and they'll be true to the type that you ordered. Rice is like that. Potatoes, russets and Yukon Gold and the rest of it. In cannabis, we don't have that. We have this sort of mixed up system. So we don't really know taxonomically, and that would be the sort of world of botanical classification, really what's happening with indicas and sativas and all that. And in the kind of human world of varieties and cultivars and strains, it's also very confusing. And I kind of put this down to two reasons. One is that science has been sort of not really applied to cannabis in an organized way because of prohibition, but also because cannabis is so highly variable, and it's been sort of dragged from one end of the earth to the other by humans doing things with it and getting different forms of it. In the strain world, and you can go into a Vancouver dispensary and see all the jars and see all the menu on the wall. There's thousands of different strains that come and go in popularity, and they have different chemical properties. And some of the time, it's a clear-cut thing that someone has a strain and that name corresponds to a plant. But there's also mix-ups in the sense that the same name will be applied to a different type of plant or a different variant on that. Or sometimes there's the same plant, but it's called different names. So there's mixing up of all that. But within the marijuana community, and certainly in the medical cannabis community, there's this kind of belief in a kind of a classification, a dichotomy, where some strains are called indica type and others are sativa type. And indica types would be, a shorter plant, shown here on the right, broader, darker green leaves, a high that's typically thought to be sort of sedative, sort of body stone, maybe more useful for things like pain. And sativa, which would be a taller, more open, lankier plant, narrower leaflets, and the high would have a stimulating or cerebral effect. And Dr. Russo last lecture sort of said you'd sort of leave it to me to comment on this. There's generally not a lot of scientific support for this sort of split of the two, but it's certainly sort of known in popular culture. I bring up this sort of show I watched last year where this guy is talking about his father says, that last shit you gave me kept me up all night. And he says, no, well, that's not going to happen. It's indica this time. And it's kind of like this sort of sedative, sleepy time body stone was important. And if you go onto a website of a licensed producer, you'll see indica dominant, sativa dominant, and hybrid, so a mix of the two. So, there's a lot of marketing and sort of suggestion that these go into a dispensary in Vancouver, and you'll see that same classification. Leafly, which is an online sort of strain review site, you can sort of select hybrids, indicas, and sativas. It's not going to go away. There's this sort of belief that it exists. So one of the things that we attempted to do, and I don't think it's the final approach to sort of solving this problem, is at least to shed some light on it, was that I worked with a population geneticist at Dalhousie University, a guy named Sean Miles, who was interested in plant diversity and the sort of the way that genomics can be used to tease it apart. And we actually got DNA samples from some diverse cannabis types, so 43 hemp types, 81 marijuana types, and we genotype them. That is, we scan their genomes for the nucleotide differences that would be shared and different between the different samples. And we found about 14,000 what are called single nucleotide polymorphisms, or SNPs, across all of those samples. And then applying, a lot of statistics and analysis, we tried to figure out if there was some groupings appearing there in the genetic types. And I mean, one of the things that was clear, and the green here is samples that were clearly hemp, in the sense that we went to the Alberta Research Council, or that's what it was called at the time, now called Alberta Innovates, I think. And they had a large hemp breeding population, and we collected leaves and isolated DNA. And that's the hemp group right there. There was one sort of outlier, which might have been, something that they thought was hemp, but might have been marijuana, or there could have been a sample mix-up. But, a lot of people look at that and say, well, marijuana is different than hemp. You knew that. You could probably look at it. But that was the first kind of real genomic evidence for that separation. But we also saw within the types that would have been labeled marijuana or drug type by the person we got the sample from, quite a bit of difference. And so we actually scored them on the percentage of what we refer to in the paper that was published, which is in PLOS One in 2015, as reported ancestry. So reported percentage sativa. So someone would say, this is 100% sativa, or this is 70, or this is 50%, or this is 100% indica, and 0% sativa. And we did see a separation of those types. So we seem to see some sort of gradient that was suggestive of sativa being, at least reported ancestry, pulling these groupings apart. One of the things too, when we looked at the sort of marijuana only, was that there was a moderate correlation between the reported ancestry, again that number is percentage sativa type, and what the genotyping said was the ancestry. So it's hard to, this is kind of glass half empty, glass half full. Some people look at that moderate correlation and say, well, then it's all total crap. Like if you're seeing somebody telling you this is 100% sativa, you can't really trust it, because there's only a sort of 0.36% chance, I guess, or that it really is. But other people say, well, that's an indication that something is happening there. And I think I fall in that second group. I think there's something happening there. And I think Ethan Russo and others are completely right in that the idea that you can look at a plant or smoke a plant and sort of say, oh yeah, I know that's, you see these numbers like, that's 85% indica. It's like, that's not really the case. But, a combination of this genotyping and more in-depth genotyping, plus the chemical analysis that others have called for, which would be, in-depth metabolite profiling that looks at terpenes and cannabinoids, can be used in combination to find what I would hope would be sort of functional groupings of the plant, where you can say this is not just chemotype, but there's something about the sort of overall combination of metabolites and the ancestry of the plant that says it's in this group. And that's sort of where we may be going with cannabis. Are we going to get to the point where we have Golden Delicious? Probably. We'll have registered varieties, and we'll be sort of beyond strains, and they'll have known properties. But people will also, there'll just be this sort of cloud or swarm of strains out there, and people will sort of want to know where they fall into. And so I think we'll be having some of these sort of reductionist, science-driven approaches. And some people scoff at that and say, well, why do you need to have a $300,000 instrument to tell you it's indica? Well, I think we need that kind of data to really tighten things up, and then maybe after that the community can sort of run forward a bit more. So a few conclusions are, going back to this biochemistry, this trichome, the focus on the trichome. I mean, all cannabis growers focus on the trichome, and it probably makes sense that cannabis scientists focus on the trichome as well. More and better genomes are needed, so we continue to do sequencing, and we do more purple kush sequencing. Genotyping analysis is starting to at least shed some light on some of the complexity of the plant, and that's also going to lead to better breeding, I think. So we're going to be able to use genomics. And one of the things I didn't do in this talk is really talk about my company, Anandia Labs, and our sort of, what we do in the commercial sense. I confine this to the science talk, but some of the stuff is that we want to use our knowledge of the genome to help create improved cannabis plants. And we're not talking genetic modification here, we're talking about breeding that can be fast-tracked with some of this information. And certainly cannabis needs more research. And I sort of turned to an article that Mark Ware, and Mark will be here in less than a month, I think, wrote for an issue in Nature Outlook last year, where we really call for sort of closing this knowledge gap, where if cannabis has been outside of university labs and company labs for decades because of prohibition, and it's been a sort of, especially in the US, a select few who have got access to the plant. If we're moving towards more access on the medical side and certainly adult use or recreational use in the future, it means we need to bring it into the labs and allow researchers to work on it, both guys like me who are basic plant biology researchers, basic science research, and people like Mark Ware and others who are clinicians who want to put it into clinical trials and figure out what makes it tick in that sense. And Canada, we say something here like Canada can be a leader, and I think it will be. We just need to still open some things up in the regs. And to acknowledge some of the people I mentioned in Anandia Labs, Jay Soller, Aaron Gilchrist, UBC Botany, where I'm an adjunct, has helped support some of these projects. I worked at NRC before, as Zamir pointed out. U of T, various collaborators, lots of funding. This was sort of a greatest hits package, so there's lots of people behind those projects. And with that, I conclude, and thank you for your time.
Thank you very much. We have time for questions. I would ask that you use the microphone so that we can record the questions and the answers. There's one over there. Feel free to ask any questions you have of John, and we'll go from there. Thanks.
I have a question about these enzymes. So the difference between a high CBD or a high THC strain would be the presence of an enzyme that makes one versus the other in an acid form. I may be wrong about this, but I think that one strain which is particularly high in CBD for medical purposes is the child of two very high THC strains. How does that work if the parents don't express that enzyme that the child would?
I don't think it's probably true. I think there's been
confusion.
Some other cross happening to create that CBD. There's been this sort of idea that hemp can wander off into the ditch and become marijuana again, and that marijuana can sort of wander over and become sort of hemp-like in its CBDA production, and it's just genetically not happening that way. The sort of hemp marijuana dichotomy doesn't really apply as much anymore because we do have high CBDA marijuana type plants, so the drug type, and so but you need to get that CBDA production from somewhere else than a pure THCA plant. I can hear you.
Is there any evidence to say that a plant that's grown with the full spectrum of light outdoors, that might change what you're talking about, like maybe be a little higher in different cannabinoids based on that full spectrum? Is there some sort of bizarre ways?
Yeah, I think what we see with plants, and the cannabis isn't the only one, is that the environment has a huge impact. So, you won't get environmental switch to make a THCA plant and a CBDA plant, but you could go from 15 to 18% and different terpene levels, and certainly a plant grown under very high sun, outdoors, dry, arid environment versus some sort of cloudy sort of rainforest is going to have quite different metabolite levels. In terms of the difference between indoor grown under high pressure sodium lights or something like that versus outdoor or sun-grown, I don't think we've seen a ton of good data that would take, you could do something like take the same clones, put 24 of them indoor and 24 of them outdoor and watch them, and then analyze chemistry and see some differences. But, it's not like, the plant is still doing well indoors, and it's got most of what it needs in terms of photons and nutrients. So it's not like it's sort of the sun is required or it's going to lack some sort of key component.
So I have a question. If you made a cross between a marijuana strain and a hemp strain, would you then get super growing fast plant that is halfway between the two species? I'm sure people have made that cross between.
Sure. Yeah, you actually get something that doesn't really perform that well as either. In the sense that, hemp typically is quite tall. In Canada, we've got some shorter hemp types because of lodging and all this kind of stuff. But hemp is bigger, marijuana can be a bit smaller, and you sort of get something intermediate in that cross. You get intermediate potency. So say the hemp variety was 5% CBDA, and the marijuana type was 20% THCA, you might get 8 or 9 or something in total, and it would be a mix of the two. So it's almost like you get there. And there's not like a ton of sort of hybrid vigor or something that results from that cross. But on the other hand, people talk about real hybrid in the drug side of things, where there's sort of disparate genotypes crossed, and the offspring will have potentially higher THCA levels than either of the parents. But again, that's a little bit anecdotal because we haven't seen that. Yeah.
I have a question.
Jeremy.
Yeah, go ahead.
So you mentioned that one-to-one strains were essentially the natural state of the plant. And that, through selective breeding, we ended up with all these really high THC strains. But now we're seeing more one-to-one strains coming back. Now, where did those come from? Were those bred, or were those fan-raised seeds that were kept in a safe somewhere and pulled out for the new plant?
Right. I think maybe I misspoke when I said the original state would have been one-to-one, because I think what it would have been is a population of sort of, outcrossing male and female mix, and this would be potentially like hashish sort of fields in Morocco or Lebanon, where you would have a mix of CBDA dominant plants, some THCA dominant plants, and then the vast majority being one-to-one, and there being this sort of like, they just harvest everything, and you would end up with this kind of material, whether it's flower or hash or something, keef or something, that would be sort of 50-50 THC, CBDA. So that's one sort of potential natural form. But there, I think, were regions where people were quite aware of the sort of psychoactivity of THCA, so they would be selecting for that, and they would be sort of growing pure stuff. But I think, to answer your question, really, like, I think these one-to-ones that we see in sort of modern cultivation would be the product of people intentionally doing that cross and then cloning plants from it to sort of maintain that one-to-one. I don't think it's well, it could be actually that they found some seed in some sort of Moroccan sample or something outside of sort of commercial production in North America and sprouted it, and it was one-to-one, and then they went from there. So maybe it's a combination of the two, but I think it's more what it is CBDA was ignored for a long time as being important, and breeding pushed CBDA out as THCA levels were rising. And it was only when things like more medical properties were being recognized, CBDA was sort of like, hey, we better bring that one back, and there's been this intentional breeding to get it back.
Yeah.
So right here?
Oh,
sorry. You control it, Samir.
Go ahead. You've had your hand up for a second. Go ahead.
Yeah.
Thank you. So I just had a question around the selection of the SNPs for the genotyping process. Were they just selected from, from more conserved regions or like clinical regions or just the entire genome?
The entire genome. I mean, we were just sort of digging deep for as much information. So it wasn't conserved, it wasn't necessarily genic regions. We could have, repeat regions, but we were, the enzymes used in the GBS method were the same. The samples were all processed by the same facility. So they were comparable across that, but those were, across the whole genome.
I'll just pass you this, make it easier.
Thank you. My question's on the upper limit of cannabinoid content. So some of the licensed producers are putting up numbers like 32% THC. I'm just wondering, what do you think is the upper limit of total cannabinoids available, like in a glandular trichome and the total plant?
Other people have asked me about this, and I was consulted by a Washington State grower a couple of years ago who was claiming this kind of mid 33 or 34%. There's one paper published from Australian samples that were sort of part of a forensic collection and maybe a sampling by the police or something, and they claim that they found some samples in the high 30s, I think, or the mid 30s. And certainly the licensed producers, some have looked at like sort of 32 or 33%, but I think this is also before they were told to adjust their THCA levels to THC levels. So, with a molecular weight of THCA being higher because of the acid group, you have to put a correcting factor, 0.877, to convert it sort of theoretically to THC. So when you do that, you naturally reduce the amount of the THCA into THC equivalents. So 33% might be 28.5 or whatever the number is. So I think there was some confusion or some variability in the industry. So we were seeing those numbers and they weren't really, they were kind of adding THCA and THC. My own take on it is I think it's kind of around 28 or 29%. That seems to be like numbers that are consistent, but again, that's in THC equivalents. My lab sees samples that are higher than 30% in THCA, but again, it's back lower once you've done the conversion.
And that limit's going to be set by just how big the glandular trichome can actually get.
I think it's like, the density of trichomes on the floral tissues, the size of the trichomes, and then what you fill them with. I guess, I think the actual sort of molar amount of terpenes is relatively low, but if you compress that further down, you might fit more cannabinoid in that compartment. But we're kind of got there now. I mean, the potency, I don't see a potency by dry weight increasing much, but potency by plant might, in the sense that you might be able to get more trichomes on more surfaces, on leaves rather than flowers and stuff, so you could get more yield that way.
Great, thank you.
Yeah. I think there was a question up top.
Thank you.
I had a question about, would it be... Thank you. I had a question about marijuana being used for harm reduction and maybe weaning off people off any type of addictions, like opioids and things like that. Is there any studies on it?
Yeah, so I preface this. I'm not a doctor. I do the plant stuff. But I attend enough conferences that I see information about opiate sparing, in the sense that there's like some sort of potentially there's the ability for cannabinoids, particularly THC, to kind of allow patients to reduce opiate, not to zero, but to reduce dosage. So I think there's lots of interesting things happening there. And I mean, the other one, you see it in the newspaper too, that in the US states where medical cannabis becomes legal, they often see a reduction in opiate overdoses. So there is something there, and people are digging in and are, I think, epidemiologically interested in the clinic as well. But it's still, I often wonder though, in BC where we have, substantial access to cannabis, not necessarily on the downtown east side, but in society in general, we're still seeing tons of opiate deaths. So, I mean, what would it be like if cannabis wasn't here, I guess, is a question. But there's a good connection there. I think needs more data.
Okay, there's a question back there.
There's the mic. Should we take
she's got it.
Yeah.
Can I ask a question?
Yeah, go ahead.
I'm just curious if there's any speculation as to why cannabis produces cannabinoids in the first place.
Yeah, I think, it actually comes back to this sort of hops-cannabis connection I mentioned in terms of the taxonomy. In that case, you have, the female flowers are where most of the trichomes are formed, and in cannabis, those trichomes contain cannabinoids, and in hops or humulus, the trichomes contain other related molecules. They don't get you high, but they're chemically kind of similar. And those are sort of like outward-facing structures, like the trichomes on the epidermis, on the surface of the plants, and they're goopy and they're sticky and they taste bad. And inside those tissues is where seed formation will occur if pollination happens. And so I think it's all about some sort of defense. And I don't think we can just sort of say like, oh, this is defense against a particular beetle that bores in there and eats. It's kind of like just bad tasting, goopy, sticky. If you've ever seen like cannabis plants that are attacked by mites or other insects, they often will be like breaking those trichomes and it oozes all over them and sort of traps them. So it's kind of like a general attempt to ward off, I think, insects. But they also, cannabinoids absorb UV light, and they have a bit of a sunscreen property as well. Some people have speculated that pollination, even though it's wind pollinated, these things might be sort of attractants for pollinators. I don't put much weight in that one, but it might not necessarily be just one thing. It's kind of like plants, to invest so much carbon and so many nutrients in those class of molecules, they got to help you in some way, and they are maybe doing a number of things for the cannabis plant, mainly defensive. Got
Okay,
one there and one
We'll
there.
take one more and then we'll break to get something to eat and drink. Go ahead.
I know you've been talking about classical breeding for the most part in your discussion here. I wonder, is there any CRISPR type activity going on in cannabis? And what might they be using it for terpenes, for cannabinoid placement or something?
Yeah, no, I mean, people are obviously interested in CRISPR. It's been used in CRISPR, just for the audience, is a sort of revolutionary genome editing technique where you can make directed changes to a genome. And it's being done in human cells and animals and all sorts of organisms. I mean, in plants, it's particularly useful for turning things off. So if there was a gene you didn't want to function in the cannabis genome, you could use CRISPR. The problem, though, is that cannabis, other than, I should say, Samir Punja's published work with genetic transformation with hemp tissue cultures, so not whole plants, cannabis hasn't been transformed in that we haven't introduced new genes into it. And some of you may breathe a sigh of relief. There's no GMO cannabis. But as a scientist, I'm like, this would be a great tool. But, I don't necessarily want it to get out into the wider world unless it's acceptable. But we don't have that tool in the lab right now, so we can't perform CRISPR. But, yeah, people are talking about it. I'm not sure we've exhausted classical breeding to the point where we need that. It's a bit of a shortcut. Yeah. Thanks.
Well, Jonathan, thank you very much for an enlightening talk, and I appreciate you coming out here. Thank you very much.
Thank you very much. Thank you. Thanks, everyone.